Welcome to On Tech Ethics with CITI program. Our guest today is César de la Fuente, who is a presidential assistant professor at the University of Pennsylvania. César's research focuses on using computational approaches to accelerate discoveries in biology and medicine. Specifically, he pioneered the development of the first computer-designed antibiotic with efficacy in animal models, demonstrating the application of AI for antibiotic discovery and helping launch this emerging field. Today, we're going to discuss the use of AI for antibiotic discovery. Before we get started, I want to quickly note that this podcast is for educational purposes only. It is not designed to provide legal advice or legal guidance. You should consult with your organization's attorneys if you have questions or concerns about the relevant laws and regulations that may be discussed in this podcast. In addition, the views expressed in this podcast are solely those of our guest. And on that note, welcome to the podcast, César.
It's great to be here.
It's a pleasure to have you. I gave you a very brief introduction, so can you share some more about yourself and what you currently focus on at the University of Pennsylvania?
Yes, absolutely. So I guess the mission of the lab is to really harness the power of machines and use that to accelerate discoveries in biology and medicine. And I'm really lucky to be able to do that with team members that come from different places around the world, but also have different backgrounds and different ways of thinking. And so just to give you an example, right now in the lab, we have computer scientists, chemists, synthetic biologists, and microbiologists all working together to try to accelerate antibiotic discovery by using a combination of AI methods and also a lot of experimental validation. So that's sort of what we're aiming to do.
So I'm excited to learn more about how AI can be used to accelerate antibiotic discovery. To get started, can you talk a bit about some of the current challenges with antibiotic discovery and development?
Absolutely. So antibiotic resistance is a huge global health problem. Just to give you a sense of the magnitude, it currently kills over 1 million people every single year in the world. And these are bacterial infections that are untreatable with conventional antibiotics. And it gets worse, actually, because the current projection is that by the year 2050, 10 million people will die as a consequence of bacterial infections, which is predicted to surpass every other major cause of death in our society, even including cancer. So we're facing a silent pandemic where a lot of these emerging pathogens are becoming increasingly resistant to all the antibiotics that we have available in pharmacies and in hospitals. And more, we see patients coming into the clinic with infections that are really untreatable, even with combination therapies. That's a huge concern and a problem that affects every corner of the world. And we're very much motivated by this. And one of the things that is quite surprising is that actually, I would say antibiotic-resistant infections is one of the most underinvested problems in the world that kills the most people. And if you look at statistics and numbers from big pharma and companies, they've over the years reduced or completely stalled or stopped investment in antibiotic discovery. And so right now in the world, in 2024, it's up to a couple of academics, crazy academics in different institutions around the world to try to think outside of the box, think about how we can push the boundaries of antibiotic discovery, how we can think about the problem in a different way, how we can develop novel approaches that are different from traditional methods to try to accelerate this and try to really come up with truly novel antibiotics that can eventually, hopefully, save lives.
Now, I want to hear more about how some of those novel approaches and specifically how AI can help overcome some of those challenges. But first, I think it would be helpful to briefly go over how AI is used in antibiotic discovery. So can you provide us with an overview of how AI is used in this context?
Absolutely. So maybe I can give you some sort of historical context and maybe threading in my own personal experience in this field. So let's go back. So when I was doing my PhD at University of British Columbia, when I went there, I was really interested in sort of understanding biology from first principles. And so I dedicated my PhD to learning about the simplest living organisms that exist, which are these wonderful creatures called bacteria. And so I learned all sorts of things about how bacteria become harmful to humans and the mechanisms that they utilize to do that. And then towards the end of my PhD, I started working with antibiotics, trying to develop small, sort of engineer small proteins to see if they could be used as a new class of antibiotics. But one of the things that I quickly realized is that biology is obviously very complex, chaotic, playful, and extremely difficult to engineer. So oftentimes I would find myself making a mutation on the molecules that I was trying to develop as a new antibiotic, and then having to go back to the drawing board. And oftentimes things wouldn't work. And I realized that, of course, biology was not programmable. So I was a bit dissatisfied by this need for endless try and error experimentation that was quite painstaking. And so then I had sort of this epiphany that with advances in compute power, I thought that AI could actually help revolutionize biology, and more specifically, maybe help revolutionize the invisible biology, which is microbiology, and also the field of antibiotics. And so then what I did is I had this opportunity. I was recruited at MIT, which back then was a mecca for AI. And there were a lot of people working on AI, but in different domains that were unrelated to biology. Things like pattern recognition systems for speech and image recognition. And at the time, the general consensus is that computers and AI could not really be used in biology because biology was just too complex for computers to have any sort of useful input. But nevertheless, I was able to absorb a lot of that information from other fantastic researchers working at MIT, applying AI to other systems. And along with our great collaborators, we decided to ask a very fundamental question, which was, can a computer be used to create an antibiotic? And again, this is the origins of this field. And so we just had to start by asking really critical questions, and really simple questions in retrospect. And so we thought about this a lot for a long time. And for example, one of the critical questions that we asked ourselves was, how can we teach a computer to innovate at the molecular level, right? To create something new, to create diversity at the molecular level. And after much thinking and brainstorming, we came up with this idea to just simply mimic the greatest engine that we have in the world to create something new, something novel, something diverse at any level. And if you think about it, that's evolution itself, the evolutionary process. And so what we decided to do is we decided to train a computer to execute Darwin's algorithm of evolution. But the really cool thing is that instead of having to wait for millions of years for a molecule to evolve through the natural evolutionary process, on the computer, we can compress that time scale tremendously. And we can do that in a matter of hours. So that's what we did. We trained an algorithm that was capable of taking natural molecular sequences from nature and was able to simply apply evolution. And of course, the primary steps of evolution are mutation, selection, and recombination. And we built a feedback loop in order to create that sort of process in a continuous fashion. And so the computer was able to create new antibiotics, entirely new antibiotics. And then it basically gave us a number of molecules that it predicted that would be great antimicrobials. But of course, this project was started around 2015 or so, and it got published in 2018. And so back then, we couldn't just rely on the computer's assumptions. We couldn't just trust that the computer was correct. We needed to experimentally validate, right, to see whether the computer was correct or incorrect. And so what we did is we synthesized chemically. We made a lot of these molecules that the computer created in the lab, and we tested them against bacteria that are clinically relevant bacteria that are problematic in our society. And we're able to find out of those screening efforts experimentally, particularly one molecule, and that was really good at killing bacteria. And we call this molecule guanine. And this was really a computer-created molecule that now was able to not only look pretty on the computer screen because it has this beautiful alpha helical structure, but it was actually capable of killing clinically relevant pathogens in the lab. Then we wanted to learn how this guanine molecule was able to kill bacteria, and we performed a critical experiment. And what we found is that it killed bacteria in a completely different way compared to most antibiotics of the same type. Basically, it targeted the membrane in the reverse way compared to most antibiotics. And this was really puzzling to us because that was in no way incorporated or written into the computer program. And looking back at that experiment, I think it really represents one of the first examples in biology of the emergent properties of AI. And these are properties that were not incorporated, not written into the fitness function by human researchers, and then realizing that the AI itself can lead to novel properties that were unanticipated. And of course, we've seen that now in many other domains, the sort of ability of AI systems to come up with new things. But perhaps that was one of the early examples in a field as complex and chaotic as biology. And then the final experiment that I think is critical to highlight is that we were able to see that guanine, this computer-made molecule, was capable of reducing infections in a preclinical mouse model. That was really the experiment that convinced us that this would be a new field. You could create something on the computer that could not only kill bacteria in vitro, in petri dishes, but also in mouse models. And so since then, this field has been growing, and we published that paper in 2018. So in the last half a decade or so, this field has grown into a more mature field and a hugely transdisciplinary field, by the way. It brings researchers from computer science, from microbiology, from synbio, all together to try to tackle this huge problem of antibiotic resistance.
That's really fascinating. So just to back up for a moment, I'm curious to hear about when you did that initial training of the algorithm, what types of data did you use?
So in that case, we use actually available data of natural molecules from nature. And so back then, we didn't have huge training sets. Right now, the latest projects that we've been conducting in my lab, we've been using more training sets. And if you want, I can tell you a little bit more about some of those projects.
Absolutely. That would be great.
Great. So that was work that we conducted at MIT. And then I was recruited at the University of Pennsylvania. And here, the initial question that we asked ourselves was, can we use computers to accelerate antibiotic discovery? And this was motivated by the fact that it takes a long time to develop a new drug, including a new antibiotic. So from the moment that a molecule is discovered in the lab to the time that it actually has an impact on patients, it takes over a decade on average. So it's a long, windy road to get to have an impact on people. And it's also a hugely expensive endeavor. So on average, it costs over $2 billion to develop an antibiotic. And just to put this into perspective, this is more than the budget that NASA or SpaceX have to take a rocket to outer space or to the moon. So it's an incredibly expensive and slow process. And so we hypothesized that maybe AI could help accelerate this so that thereby reducing the time and the cost associated with discovery of antibiotics. So that was the initial premise. And so, we were asking ourselves, how can we go about this? And so through some of the technologies that I had learned back at MIT, the pattern recognition algorithms that I mentioned earlier, we decided to apply those algorithms for antibiotic discovery. But of course, instead of recognizing facial expressions or sounds, we wanted to recognize molecular patterns, right? Code, biological code, to see if we could find potential antibiotic molecules in biology. And by biology, I mean things like genomes and proteomes. Genomes being all the genes that are expressed in a particular organism, and proteomes are all the proteins that are expressed or encoded in those genes. And so what we did is we started with very simple pattern recognition algorithms that could mine individual proteins to see if we could find antibiotic molecules encoded within those proteins. So this was in collaboration with colleagues over in Italy. And we performed this work in a number of projects, mining individual proteins. But then we realized that with advances in compute and algorithmic power, we could scale up from mining individual proteins to mining entire proteomes. And these are all the proteins encoded in a genome. So we decided to embark on this very exciting journey of, for the first time, mining the human proteome as a source of antibiotics. And when I talk about the human proteome, typically we think of 20,000 proteins that are encoded in our genome. But in this case, we also took into account isoforms. So in the end, we sampled with the algorithm over 40,000 proteins, which if you do a calculation, it corresponds to around 100 million peptides. This is obviously a huge endeavor at the time. And so we thought that the algorithm would take a while to process through all this data. And to our surprise, within an hour of the onset of the project, the algorithm stopped and it finalized the task. And so initially, this was very surprising. We thought that there had been maybe an error in the algorithm. We went back to the code, there was no bug. And we simply realized that the algorithm was actually a pretty simple scoring function that was very computationally inexpensive. And so it was extremely rapid. So in just 1 hour, he was able to sample the whole human proteome. And what this work led to was the discovery of thousands of new, what we call encrypted molecules, encrypted peptides that were encoded in the human body that were previously unrecognized to play a role as antibiotics. So this is sort of dark matter that was previously unknown to have this antibiotic property. So this really opened up an entire new field of mining dark matter to try to find potentially useful molecules. And so then we synthesized a lot of them and we validated them, and many of them had actually anti-infective properties in preclinical mouse models of infection as well.
I want to take a quick break to tell you about CITI program's bioethics course, which provides an overview of bioethics concepts, principles, and issues. For example, it addresses clinical ethics, genetics, justice in healthcare, and more. You can learn more about this course at citiprogram.org. Now, back to the conversation with César. From your perspective, what do you think these findings may mean for the future of antibiotic discovery and development?
If I look back, I think a lot of this AI work and algorithmic work, what it has enabled, and I can talk about subsequent projects that we followed up on, it has enabled us to accelerate discovery of antibiotics. So if you think about traditional methods, it takes between 3 and 6 years to discover preclinical candidates that might be exciting to take to phase 1 and phase 2 and phase 3 clinical trials. And now with AI, we can do that in a matter of hours, but we can actually discover hundreds of thousands of preclinical candidates. And so we basically compress the time that it takes to discover potential antimicrobials from years to a matter of hours. And I think that's really one of the takeaways that, if I reflect back on the work that we've been doing, it's one of the most surprising things. If you had asked me maybe 5-6 years ago, I would have probably said that's close to impossible. But with advances in compute and our ability to develop better and better algorithms and to mine entire genomes and entire proteomes, to really mine, explore biology as a source of molecules and potential medicines, this has been really transformative.
Absolutely. So just to shift gears for a moment, when it comes to these ongoing efforts in antibiotic discovery, what are some of the unique ethical and legal issues that you and your colleagues are considering as you do this work?
Yeah, that's a great question. And I'll tell you a little bit about another project that really ties in well with bioethical and other potential ramifications. And it's a follow-on work from the exploration of the human proteome. So when we explore the human proteome as a source of antibiotics, we found, like I mentioned, a lot of these encrypted sequences, right, that were previously unknown to have antibiotic properties, and they were hidden messages in our own bodies that we didn't know about before. And so that led us to hypothesize that most likely we would find similar sequences or similar molecules across the tree of life, not only in the human body, and throughout evolution, that maybe we would find similar things, right? And since we had done the mining of the human proteome, we decided to next look at our closest relatives, which are Neanderthals and Denisovans, to see if we could find similar molecules encoded in their genomes and their proteomes. And so in order to tackle that, we had to develop a machine learning model. It was a machine learning model that, in broad terms, was able to discover antibiotic molecules encoded in Denisovans and Neanderthals. And this was quite exciting, right? It was the first description of any sort of therapeutic molecule discovered in extinct organisms. And we call this new field molecular de-extinction. So the notion of bringing back molecules from the past to address present-day problems, such as the problem of antibiotic resistance. And one of the motivations was that perhaps molecules that existed hundreds of thousands of years ago could be brought back, because one of the rationales is that bacterial pathogens today would have never encountered those molecules. So maybe they give us a better chance of fighting off contemporary pathogens. And so we did that, and it was truly an exciting moment, but it was the time to actually resurrect those molecules using chemistry. And because as far as we know, some of them at least, they're not expressed in living organisms. So they're truly molecules from the past, as far as we know. And so we really resurrected them using chemical methods. We brought them to life, if you will. And then we tested them against bacteria, and some of them were active against contemporary pathogens, not only in vitro, but also in preclinical mouse models of infection. So this was really exciting, and it convinced us that we could sample extinct organisms as a source of antimicrobials, as a source of potentially useful molecules. And so what we did next is that we just challenged ourselves, and we said, why don't we just sample every extinct organism known to science instead of just Denisovans and Neanderthals and modern humans? And we decided to mine what we call the extinctome, which is essentially every extinct organism known in the literature. And in order to tackle this, we had to develop a more powerful machine learning model. So we developed a deep learning model that enabled us to mine hundreds of proteomes at a time. And this work was extremely exciting. It really allowed us to discover new antibiotics in creatures from the past, including the woolly mammoth, the ancient elephants, and some ancient penguins, and even a giant sloth that has a very interesting history. It was actually the remnants of this giant sloth were initially discovered by Charles Darwin in one of his expeditions to Patagonia. So we're able to find a whole new world of antimicrobial molecules that were really previously just hidden in plain sight in all these extinct organisms. And I'm talking about organisms going from the Holocene to the Pleistocene. And so basically, we brought those molecules back to life, and many of them have anti-infective activity in preclinical mouse models. And so we now have a bunch of preclinical candidates coming from all that work. But going back to the initial question of bioethics, from a bioethical standpoint, one of the things that worried me a little bit is throughout this molecular extinction work was what does it mean to bring back a molecule that used to exist, hundreds of thousands of years ago, back to the present world? Is that okay to do that? And so from early on, we've been consulting with bioethicists to make sure that whatever we do in the lab, we do it responsibly. And we truly believe, as one of the ethos of the lab, about responsible innovation. And so one of the things that we've been doing, for example, is whatever molecule we synthesize chemically to make sure that it cannot self-replicate. And, if it were to escape from the lab, it would not be able to self-replicate in any way. And we've also been obviously storing every molecule that we synthesize very securely and safely. And so that's something from a bioethical perspective that we take up very seriously. And we've been talking to people that know a lot more about bioethics than us, that can really advise us as to how to do the science that we do in the best way possible. The other thing that maybe I'll mention that I thought it was pretty funny is that when we were initially discovering all these new sequences, new molecules from extinct organisms, I went to the patent office here at University of Pennsylvania, and I had this kind of funny conversation. I told them, I know that natural sequences are not patentable, right? Because through the myriad case, they were deemed not to be patentable in natural sequences because they were produced as products of evolution. But I asked them, what about extinct sequences or extinct molecules that sort of existed tens of thousands of years ago, but are no longer present in the living world, in the natural world that we live in? And they looked at me like I was crazy, and they didn't know what I was talking about. So I explained a little bit more on the project, and then basically they don't know. So this is creating a new area of patent law, where patent lawyers are trying to figure out whether molecules from the past might be patentable or not. And so I've actually written an opinion piece with a patent lawyer, an expert in the field, to try to provide a balanced view as to the pros and cons of patenting this sort of molecules from extinct organisms.
So given the novelty of this work that you're doing, which again is very fascinating, and also some of the issues that it's raising, like you were just talking about with patentability, do you have any additional resources that you suggest where people can learn more, or perhaps even just advice that you have for folks that might be looking to also get into this type of work?
Yeah, these are all really emerging areas. I would say 5-6 years ago, AI for antibiotic discovery was not even a field. Molecular extinction is something tangible as of a year ago, but people can refer to our website, and I also have LinkedIn and X accounts, and we try to report some of our latest findings there. But I would say, I would really encourage young minds, if they're interested in a problem of global magnitude that affects every corner of the world. I think antimicrobial resistance is such a problem. It's one of the most underinvested areas that you can find in our society that actually affects and kills the most people. It does not discriminate. It affects people everywhere in the world that you care about, that you think about. And it's predicted to get much worse. And then I would just like to highlight the importance of antibiotics. Antibiotics are not only helpful to treat an infection when we get infected and they save our lives, but they're also critical for modern medicine as we know it. So things like even childbirth or chemotherapy treatments where patients, for example, are immunocompromised, oftentimes they can die as a consequence of infections, right? And so you need effective antibiotics to treat those patients. In surgeries, you need effective antibiotics. And in every critical and routine procedure in modern medicine, you need to have effective antimicrobials. And so they're really sort of treasure medicines that we need to value tremendously. And without good antibiotics, we face an uncertain future where we might go back to a pre-antibiotic era where people could just die from a simple scratch that gets infected. And also, I think it's important to underscore that, over the last 100 years or so, humans, we've been able to almost double our lifespan. And this is really thanks to three main pillars: antibiotics, vaccines, and clean water. And so imagine removing one of those pillars. We will be facing a really uncertain future where infections could really have a dramatic effect in our society.
Absolutely. And going back to the resources that you mentioned, I'll certainly include links to those in our show notes so our listeners can learn more and follow you and connect. So I guess my final question for you then today is, you mentioned earlier about how we're increasing our abilities with compute power and also algorithmic design, and also these new fields that you've helped forge are advancing. So where do you see this field headed given those things?
I think what I'm most excited about, and I think going back to the young minds, whoever is listening, we really need people, young talent, driven talent, wanting to change the world to actually join some of these efforts. But I think what I'm dreaming about at the moment is that we will be able to use AI to really discover new antibiotics that can enter the clinic right away. And in order to do that, we need to develop algorithms and not only optimize for antimicrobial activity, so to kill bacteria more effectively, but also they need to be optimized for all the other parameters that make a drug. And this includes having molecules that are non-toxic against human cells, that are stable enough to be able to accept their antimicrobial function before they get degraded. That we need molecules that can distribute throughout the body and reach the infection site, in fact, be able to clear the infection, the reduced off-target effects as much as possible. And so we are developing such parts of the toolkit, different algorithms that can optimize for each of those different properties that you actually need to build a drug. And in the future, perhaps we'll have an Amazon type system for personalized medicine. Let's say if a patient has an infection caused by a specific bacterial pathogen, maybe there's a rapid diagnostic that tells us, okay, this is the pathogen that needs to be targeted. And perhaps then an AI coupled with automation can quickly create an antibiotic specifically for that patient, specifically for that infection that is causing problems in that patient. So maybe that's an imaginary future right now, but maybe we'll see that someday.
And that is a great place to leave our conversation for today. So thank you again, César.
It's been a pleasure. Thank you, Daniel.
I also invite everyone to visit citiprogram.org to learn more about our courses and webinars on research, ethics, and compliance. You may be interested in our Technology, Ethics, and Regulations course, which covers various technologies and their associated ethical issues and governance approaches. And with that, I look forward to bringing you all more conversations on all things tech ethics.